Hello my dear students, you are welcome. In this video we are going to revise paper 1, cell membranes and transport, part 1. Let's start. What is the correct range of measurements for the width of the cell surface membrane? Letter E 0.5 up to 1 nanometer, letter B 5 up to 10 nanometer, letter C 50 up to 100 nanometer, letter D 0.5 up to 1 micrometer. Of sure letter D is wrong because the unit here micrometer. We measure cell surface membrane by smaller unit called nanometer. It's okay. Look firstly at this figure. This figure taken by transmission electron microscope. It shows two dark lines, and in between these two dark lines lighter area, and this three laminar part represent phospholipid bilayer, okay, where tails, hydrophobic tails, represented by this lighter area, while heads of phospholipids from above and from below. Look second figure like this one. Here the dimension or the width of cell surface membrane by its phospholipid bilayer around 5 nanometer. So your answer is 5 to 10 nanometer, and with average equals 7 nanometer. 7 nanometer. Okay, number 1. Number 2. Where in the cell surface membrane are the carbohydrate chains of glycoproteins and glycolipids mainly located? They are mainly located where? Precisely where? Look at this figure, cell surface membrane, this carbohydrate part red in color, okay, attached either by protein or attached with lipid, glycoprotein and glycolipid. They act as receptors for hormone, certain hormone. This hormone act as cell signaling, okay, so their location must be on the outer surface. Sorry, on the outer surface. So the answer is D, okay, the answer is D, for receiving cell signaling molecules like hormones, for example, like insulin, like glucagon, and so on. Number 3. Sodium ions can enter cells across the cell surface membrane. Which methods could be used by sodium ions to cross cell surface membrane and enter a cell? Sodium ions like Na+, it's simple Na+, it is charged. It is electrically charged, complete charge. So when sodium ions pass through cell surface membrane, it passes within certain gates called channel, protein channel, like this channel. This protein channel is specific for sodium ions only, okay. It passes through this channel, okay, from high concentration to low concentration. It obeys what? Diffusion. But here by the help of what? Carrier protein or protein channel. So it obeys diffusion, okay, diffusion principle, from high to low, down concentration gradient across membrane by the help of this channel protein or carrier protein with specific shape fit with the sodium ion shape, okay. So this is called facilitated diffusion. This is the first method to allow sodium to pass through. What is the second method? Second method, like in sodium potassium pump in cell surface membrane of all cells, of all animal cells, for example. Look please to this figure. Here sodium ions passes by certain carrier protein found within the cell surface membrane. It has binding site specific for sodium ions to bind with. Okay, how this molecule of carrier protein changes its shape by the help of ATP, energy supply released from cell respiration. So this active uptake, this active transport done by the help of ATP. So sodium passes by two methods, active transport and facilitated diffusion. Then the answer is B. Okay, all ions passes by this method, okay, through channels, okay, ion channel specific for this ion, and or by a certain mechanism need energy like active uptake or active transport. Say for example nitrate from soil passes by these two methods. Okay, what about active transport also? Active transport, the ions passes from area of low concentration to area of high concentration against concentration gradient. Okay, it against concentration gradient. Number 4, the diagram shows how an artificial partial permeable membrane was used to separate 5% sodium chloride solution and 10% sodium chloride solution in a beaker. The two sides of the beaker were labeled R and S. R containing 5% sodium chloride solution, S containing 10% sodium chloride solution. They are different in concentration of solutes, so they are different in water potential. Which one with higher water potential? That contain less solutes, so this high water potential, here low water potential. Look at the osmosis, but before the figure, look at the table and the question. Which row correctly describes and explains what will happen in the half of the beaker labeled S? Labeled S, after a while, after time progress or time pathing, what would happen to S? Water level or solution level increases. Okay, increases. What about the level or the volume of R? Decreases. He asked about S. S well increases, so the solution volume of solution increases. The answer A or B, either A or B. What about explanation for this process? Net movement of water from a higher water potential to lower water potential. The water move in this side from high to low. Look at this animation. Here represent, say for example, R. Here represent S. Okay, here less solutes, here more solutes. Look at water pass through partially permeable membrane, allow S to rise and R to drop in its volume. It's okay, so the answer is A. Number 5. Agar cubes can be used to demonstrate the effect on diffusion of changing the surface area to volume ratio. 3 different agar cubes made using a colored indicator solution were placed into dilute acid and diffused into the cubes. As the acid diffused into the agar cubes, the color of the indicator changed. Okay, the cubes had volumes of 1 cubic centimeter, 2 cubic centimeter, 3 cubic centimeter, different volumes, and were left in the dilute acid for 10 minutes. All the other variables were kept the same. After 10 minutes, the agar cubes were removed from the dilute acid and cut into half. The cut surfaces were observed and the results were recorded as diagrams. All diagrams were drawn to the same scale. The result for the 2 cubic centimeter are shown. Okay, Look, which diagrams show the results for the 1 cubic centimeter and 3 cubic centimeter relative to 2 cubic centimeter in the diagram. Okay, to this part. Firstly, the experiment like this one, but not exactly. Here, 4 agar cubes, okay, with the same volumes, here with different volumes, no problem. Here, how we made this agar jelly? By making or dissolving agar powder with sodium hydroxide solution in presence in phenolphthalein. The color will be red in color like this one exactly. Then after hardness of these cubes, it placed inside beaker containing dilute hydrochloric acid and left to stand, okay, for certain time. What would happen? The acid penetrates or diffuses inside the 4 cubes with the same rate. Why? Because they are with the same volumes, and decolorizes the color of phenolphthalein as it neutralizes sodium hydroxide, and all the cubes became what? Colorless at the same time period. Here in this exam, in this question, 1 cubic centimeter, 2 cubic centimeter, 3 cubic centimeter. Expect here with 2 cubic centimeter, the answers, look at the figure A, B, C, and D. Of sure, like this one, larger, medium-sized cube, and smallest cube like this. Look at the diffusion distance where acid neutralizes the alkali. Okay, of sure letter B is wrong. Why? Because letter B here for 3 cubic centimeter, it shows still only this part is still colored with red. This is wrong. It must be bigger or larger in redness, larger in redness around the center. So B is wrong. Also D is wrong. So you can choose between A and C. The most logic according to the result here in or for 2 cubic centimeter was C. The answer C. It's expected to still certain color still in the 1 cubic centimeter, okay, and for 3 cubic centimeter with larger red area and smaller diffusion distance. Okay, so the answer is C. Number 6. The diagram shows part of cell surface membrane. Which row correctly identifies the molecules labeled 1 and 2? 1. Glycoprotein, right. Number 2. Phospholipid head, phosphate head. So the answer is C. The answer is C. According to this figure. Here glycoprotein. Okay, glycoprotein like this one, and the glycolipid like what? Like this one, for example. This or this one. Number 7. The diagram shows the mean dimensions of an epithelial cell in micrometers with 3 dimensions. Which row shows the surface area to volume ratio of the epithelial cell, and how the surface area to the volume ratio of the cell would change if the cell width doubled? Cell width doubled. What would happen to surface area to volume ratio? To calculate surface area, here is this rectangular shape has 6 faces. Each 2 faces are different from 2 others. So surface area for this face, for example, okay, like this one and this one. okay, equal to two sides times 3, okay, times 2 equal 12. Then 2 times 8 times 2, this part 8 here from this part and this part, okay, so 2 for 2 faces times 8 times 2 the width of each face, okay, equal 32. Then last two faces, 2 times 8 times 3, so the total surface area for all 6 faces equal 92. What about the volume? The volume is easier and simpler, where volume equals 3 dimensions, equal what? Equal 2 times 3 times 8 equal 48. Okay, the ratio now between surface area and the volume, 92 to 48. By common factor, divide 4 in both sides, we reach 12 and 23, or 23 to 12. So the answer C or D. Here he asks what would happen to the surface area to volume ratio if the cell width, would change if the cell width doubled, I mean become 4 micrometer. What would happen? As width increases, surface area to volume ratio decreases. So the answer C. Okay, the answer is C. Number 8. Which diagram shows the correct direction of net water movement between the 4 cells due to osmosis? The rule of osmosis, the more negative value of psi of water potential means what? More negative value, more figure, but with negative sign means less water potential. Means also what? So it receives water from a less negative value. So for letter A, check letter A. Minus 40, more negative value. More than what? More than this one, more than this one, more than this one. So it receives water from the adjacent right side cell with value minus 22, it receives water. Also from minus 36, it receives water because it has lesser water potential on both than both. Look at minus 33, passes water by diffusion, passes water by osmosis to the cell. Okay, also minus 22 passes water to what? To minus 33 and the minus 36. So the answer is A, is right. Okay, the answer is A. Number 9. An investigation was carried out into the effect of 4 different treatments on the permeability of cell surface membranes and tonoplasts of beetroot cells. Beetroot cell vacuoles contain a red pigment. The pigment cannot diffuse through the tonoplast or cell surface membranes. 1 cubic centimeter cubes were cut from beetroot tissue and washed in running water for 20 minutes to remove any pigments released from damaged cells. 2 cubes were then placed in each of 4 test tubes containing different contents and observed for 5 minutes. What are the different contents here? The first test tube containing dilute hydrochloric acid, second one ethanol, third one water at 20 degrees Celsius, water at 80. The observations, we asked to match the observation with the treatment and its explanation. Which row shows correct explanation for the observation recorded for one of the treatment? Which one is correct? A, B, C, or D? When beetroot cells or tissue, okay, or cube put inside dilute hydrochloric acid, the observation contents of the tube stay clear. Explanation: membrane proteins have been denatured. This explanation means what? Denature of protein means damage of cell surface membrane or tonoplast of vacuole. So how come this observation obtained? It must be colored with red. So this wrong choice. What about ethanol? Ethanol is organic solvent. It is successful solvent for many organic matter, especially lipids, grease, phospholipids. Here contents of the test tube turn red, right? Why? Because ethanol is organic solvent, can dissolve lipids including cell surface membrane or membrane phospholipids. So the answer is B. The answer is B. But we need to check. Alcohol is an organic solvent, can dissolve lipids and phospholipid. So the answer is B, right? Let's check letter C and D. If we place beetroot in 20 Celsius degrees, contents of test tube stay clear, right? Membrane proteins have been denatured. How come? How this treatment caused denaturation of cell surface protein? How come? This wrong explanation. Stay clear because the cell membrane is intact, is healthy, okay, not affected by water at 20 Celsius. What about at 80? Contents of test tube turn red, okay. Lipids including membrane phospholipids have dissolved. Hot water unable to dissolve fats, like when we wash our hands after having a meal full of fat or greasy. What would happen by hot water? Hot water only unable to dissolve fat from our hands. We need soap, we need detergent to dissolve this grease or fat. So this wrong also, okay. The answer is B is correct. Number 10. Phospholipids are formed in a similar way to triglycerides. A sample contains 6 phospholipid molecules. The molecular weight of the phosphate ion is 95 grams per mole. The molecular weight of each individual fatty acid in this sample was found to be 282 grams per mole. The molecular weight of glycerol is 92 grams per mole. The molecular weight of water is 18 grams per mole. What is the molecular weight of the sample in gram per mole? We asked to calculate the MR of phospholipid, okay, sample containing 6 phospholipid molecules. Step by step. This phospholipid molecule containing phosphate head, polar head, phosphate group, and 2 fatty acids. Okay, 2 fatty acids. This is just diagram of phospholipid molecule. Look at the first step. To calculate the MR, one phospholipid molecule means containing 1 phosphate ion plus 2 fatty acids plus glycerol. Then 1 phospholipid molecule means 95. Okay, we compensate all these figures in this formula. 95 plus 2 times 282 because 2 fatty acids plus 92 the molecular weight of glycerol, okay, 92. So the total equals 751. That's for one phospholipid molecule. What about 6 phospholipid molecules? 6 phospholipid molecules equal 751 times 6 equals this figure, 4506. Okay? Then, what about water? Where is water here? During synthesis of phospholipid, okay, we synthesize this phospholipid by condensation reaction, by removal of water molecules. But how many water molecules are removed to synthesize one phospholipid molecule or 6 phospholipid molecules? How many? Look first. Here the simple form glycerol, okay, and 2 fatty acids. Here first fatty acid, second fatty acid joined with glycerol in purple color, CH2OH, CHOH, CH2OH before condensation. Look at this orange bond. This orange bond represents the site of condensation reaction, where we condense or remove water molecules. Look at this one. Here the first water molecule is removed. Here second water molecule removed. The third water molecule is when glycerol with the third carbon atom joined with phosphate group. So how many water molecules are removed? 3 per one molecule of phospholipid. First, 2, second, third molecules of water. Okay, so the calculation now, number of water molecules removed by condensation equal 3 per molecule. But here, how many molecules are needed in this sample? 6. So times 6 equal 18. Okay, mass of water removed, 18 times 18. 18 times 18 equal 324 gram, 324 gram. Okay, last step, molecular weight of 6 phospholipid molecules equal this figure minus this figure to obtain 4182 gram per mole, like exactly letter B. Okay, the answer is B. Number 11. Which statements about phospholipids in cell surface membranes are correct? Fatty acid tails allow most ions to pass through the membrane. Fatty acid tails, they are hydrophobic. How they pass ions? This wrong. This wrong statement. Oh sure. Look, this phospholipid bilayer with tails, fatty acid tails like this part and this part. How they allow most ions to pass through? It is wrong statement. So number 1 is wrong. Okay, wait. This wrong one. Hydrophobic tails point inwards facing each other. Yes, they are facing each other. Okay, this statement is right. All polar heads face the cytoplasm. Here polar heads face cytoplasm, right, in this part. What about the other layer? Here this heads faces tissue fluid, extracellular. So this wrong. So number 3 is wrong. Not all faces. Okay, the phospholipids help with the flexibility of the membrane. Oh sure, they are flexible due to what? Bending of fatty acids. Okay, hydrophobic interaction between them, also presence of cholesterol within molecules of phospholipid. This the membrane also called fluid mosaic model. Fluid, it must be flexible, okay, and change its shape or orientation of phospholipid bilayer. So the answer 2 and 4 together, 2 and 4 means letter D. Okay. Number 12. In an experiment, pieces of onion epidermis are put into 3 different concentrations of sucrose solutions B, Q, and R. The pieces of onion are left for an hour and then examined using the low power of a light microscope. Each diagram shows one cell from the epidermis that was placed in each of the sucrose solutions or concentrations B, Q, and R. What explains the appearance of cells in solution Q? Letter E, the concentration of solution Q is equal to the concentration of solutes in the cell sap. This statement is incorrect, where when we deal with net movement of water, we deal with water potential. We state that water potential, the tendency of water movement from high water potential to low water potential. But when we deal with concentration of solute, it's misleading to express the term osmosis. So letter A is wrong. Letter B, the cytoplasm has the same concentration of sucrose as solution Q. Cytoplasm is part of the cell, but the main factor that affect osmosis in plant cell is a vacuole. Okay, so the term B is incorrect. The driving force of osmosis is the water kept or stored inside the vacuole. The vacuole in plant cells, especially epidermis of onion, it may occupy most of cytoplasm. About 90% of the cell volume is a vacuole. Okay, but letter B tell us about cytoplasm. Okay, so letter B is wrong. Letter C, the water potential of the cytoplasm is equal to the water potential of the vacuole. This statement describes the internal state of the cell, cytoplasm, and the vacuole. But what about Q, the external environment where cells lie? So this also is incorrect statement. What about D? D, the water potential of the cell sap, the protoplasm, not the cytoplasm only, but all the content inside the cell membrane, is equal to the water potential of solution Q. This statement is correct. It describes osmosis. It describes the net movement of water in and out of the cell is equal, okay, in solution Q. Okay. Number 13. The electron micrograph shows rod-shaped bacteria. The actual length of the bacterium is 2 micrometer and the diameter is 0.5 micrometer. Assume that the bacterium is cylinder shaped. What is the surface area to volume ratio of the bacterium? Firstly, in this question, we need to deal with cylindrical shape, volume, and surface area. We use 2 formulae like this one. For surface area, we sum the surface area of the circle and this one in the formula 2 pi r square. Then we add 2 pi r h for the rest of the cylindrical shape. Okay. Now for the volume, pi r squared h, pi r squared h. Firstly, surface area. By compensate the figure, 2 pi r squared, okay, plus 2 pi r h, where h is the length of the bacterium, 2 micrometer, equals this figure. Please don't approximate or round now. For volume, volume equal pi, okay, r squared h, where h 2 micrometer, equals this figure. Now is simpler step, surface area to volume, both figures put together, and pi dividing the smallest on both sides, we obtain 9 to 1. So the answer is D. Okay. Number 14. Before mitochondria are extracted from cells for microscopy, they are usually kept in 0.25 mole per liter sucrose solution. Why is the sucrose solution used? Extraction of mitochondria or any other organelles from any cell depend on certain technique called cell fractionation. Okay, it depends on certain device called centrifuge, where we allow centrifugation for samples of homogenization or homogenized tissue, any tissue, okay, for certain rounds per time, okay, per speed of the centrifuge. We obtain different pellets at each time. We obtain certain pellet and supernatant. The pellet firstly, the first pellet obtained, the heaviest one. The last pellet obtained is the lightest organelle. The heaviest organelle nucleus, in order nucleus, chloroplast if we deal with plant tissue, okay, then mitochondria, then lysosomes, then ribosomes, in order. Now the question about mitochondria and about why we use certain concentration of sucrose solution. This sucrose solution, firstly, to act as solvent. We can use any other solvents. Why specific concentration of sucrose? Okay, why? Of sure, the answer is C, to prevent the mitochondria from changing in dimension, where the mitochondria itself contain matrix inside with chemicals, with enzymes, with compounds. All of these compounds and enzymes need specific water potential and certain concentration of solutes. So to keep the mitochondria intact during study, we have to extract it from tissue homogenate safely, okay, and in intact form. So we use this concentration, specific concentration of sucrose. The answer is C. Let's break down the other incorrect choices like B and D. To enable the rate of reaction of mitochondria to be determined, this later on, not during extraction, we can use certain solutions, certain chemicals, certain ingredients to check the rate of reaction of mitochondria to be determined, not for microscopy, not for viewing on stage of microscope, I mean electron microscope, or even light microscope with certain high resolution power. Last choice, to provide a source of energy. In this study, when we deal with extraction, we use sucrose solution with specific concentration not to feed or for nutrition of mitochondria. No. Okay? It is used as medium with the same water potential like matrix to prevent changing its dimension or its shape. So the answer is C. Okay? Number 15. The photomicrograph shows a type of blood cell. A type of blood cell. Of sure, this red blood cells. Which statements about this cells are correct? Oxygen diffuses through the phospholipid bilayer. Number 2, sodium ions diffuse through the phospholipid bilayer. Number 3, water passes in and out of the cells by osmosis. Let's check one by one. Number 1. Firstly, this figure shows certain molecules, transported molecules to be transported inside the cell. For example, phospholipid bilayer here. If molecule passes directly through phospholipid bilayer, it is simple diffusion from high to low. if this molecule need certain carrier protein or protein channel to pass through, it is facilitated diffusion. Both simple diffusion and facilitated diffusion is passive transport, follows obey concentration gradient from high to low. What about molecules passes or moves from low concentration area to high concentrated area? Like this molecule, it needs energy, okay, to push, to drive what? This pump protein or carrier protein to be changed in its 3D shape, conformational shape, to allow this molecule to pass from low concentration to high concentration against concentration gradient. This active transport. Another figure. Look, what about gases? Gases passes through phospholipid bilayer, although the tails of this fatty acids within the membrane is hydrophobic. Gases diffuses easily without permission, without asking. Why? Because they are hydrophobic. The gases O2, like this molecule, and CO2, like another one, passes in and out of the cell membrane, okay, easily from any area, okay. It doesn't, or it don't need, these gases don't need any carrier, any gates, any channel. It passes easily by simple diffusion, okay. What about water? Water move in and out or through cell surface membrane depending on osmosis, depending on water potential, but it needs certain gates called aquaporins, okay. Aquaporins, although it is small molecule, or because it has small structure, tiny molecular structure, it can pass easily also through phospholipid bilayer, okay. This exception for water. What about for glucose? Glucose need gates or channel, like GLUT protein within the membrane of cells to allow glucose to enter the cell, okay. Why? Glucose is polar molecule. Polar, why? Because it contains a lot of OH in its structure. It dissolves in water and passes through certain channel, okay. The interior of this protein channel is hydrophilic, okay. What about ions, positive or negative ions? Passes either by facilitated diffusion, like sodium channels, like chloride ion channels, like calcium channels. This channel is specific for each ion, okay. This facilitated diffusion. But sometimes if this ions obliged to pass or to move from area of low concentration to area of high concentration against concentration gradients, it need energy from the cell respiration in the form of ATP, okay, like sodium potassium pump. So ions move by both methods, facilitated diffusion and active transport. What about steroids? Steroids, it's derivatives of lipids. It is fatty, it is hydrophobic, so it passes easily through cell surface membrane, in and out, or through the cell surface membrane, because they are hydrophobic in nature, okay. This all molecules we discussed now, all molecules, how they pass through the cell surface membrane. So check now. Number one, oxygen diffuses through the phospholipid bilayer. Yes, right. Sodium ions diffuse through the phospholipid bilayer. No, it is incorrect. So two incorrect. Number 3, water passes in and out of the cells by osmosis. Yes, so 1 and 3 together, so the answer is C. Okay. Number 16, which row is correct for parts of phospholipid molecules? Can be saturated or unsaturated. The term saturated or unsaturated is applied to tails of fatty acids, so the answer B or D. Okay, look at this figure. Triglycerides, glycerol plus 3 and 3 fatty acids joined together. Phospholipid, phosphate head plus 2 fatty acid tails. Okay, 1 fatty acid is replaced by phosphate group, okay, and joined with a glycerol instead of the third fatty acid. Look please step by step. Let's compare by chance. By this way we can compare between triglycerides and phospholipids. Point of comparison: function, energy storage for triglycerides. It present as oil or fat in fatty tissue, in adipose tissue, for long-term energy reserve. Phospholipids, structural component of cell membrane. The main component of cell membrane is phospholipid. Number of fatty acid tails: 3 fatty acids like here, 2 fatty acids for phospholipid. Contains phosphate, no phosphate in triglycerides, while phosphate group head present in phospholipid molecule. Polar or non-polar, triglyceride entirely non-polar, hydrophobic molecule, while phospholipid is hydrophilic due to presence of this phosphate group head. It has polarity. Okay, this structure: glycerol and 3 fatty acids, glycerol 2 fatty acids, and phosphate head. Okay, this is formed by condensation process. The bond between the 3 fatty acids and glycerol is called ester bonds, 3 ester bonds, while here 2 ester bonds, and the third bond is covalent bond between glycerol and the phosphate group, also made by condensation reaction. Now return to the question: which row is correct for part of phospholipid molecule? Can be saturated or unsaturated is term related to tail, to fatty acids, so the answer B or D. Can also be found in triglyceride, also be found in triglyceride. Okay, sure. Here tail also can be found in triglyceride, tail also. Head, this part not found in triglyceride, so head is wrong, while tail is right for both, so the answer is D. Okay, number 17. A student filled dialysis tubing with sucrose solution and knotted both ends. This formed a cylinder with a length of 5 cm and a radius of 2 cm. What is the surface area to volume ratio for this cylinder of dialysis tubing? Firstly, the same 2 rules. We dealt with these 2 rules before in the same video. For surface area, we can also use this formula: 2 pi r times h plus r. We can use this. Okay, we used this before. Now for surface area of this tubing, dialysis tubing, equal to pi r square plus 2 pi, okay, r h, like here. So the total surface area equal 87.92. What about volume? Pi, okay, pi r square times h, so equal 62.8. Surface area to volume ratio equals this both figures. To simplify both, divide on the smallest. I mean here divide 62.8 to obtain what? To obtain this figure 1.421. So the answer is C. Okay, the answer is C. Thank you for watching. Hopefully you find a little benefit from this video. Best of wishes for all of you. Bye bye.